Good morning. Can you see the slide?
Yes, sir.
Okay,
Yes, sir.
very good. Thank you very much. Today we shall start lecture 8, and the topic is stilbenes and flavonoids. Already flavonoids we discussed in our earlier lectures. Today I shall review a little bit, but stilbenes, they are another group of bioactive natural products, and they are phenolic compounds, and they are industrially as well as medicinally very important. So today we shall discuss the stilbenes, their properties and structural diversities. We shall discuss classes of flavonoids as well as their medicinal activities and other biological principles. So today we shall discuss stilbenes, chemistry, biological significance, commercial uses, pulp and paper problems, because stilbenes, they are very important in paper and pulp industries, because they are problematic for making the paper and pulp from the plant materials. Flavonoids, we shall discuss a little bit about the chemistry, a bit review the earlier class, and biological significance, why they are biologically important, and commercial uses also, uh, and how flavonoids are important in pulp and paper problems. And we shall also discuss on condensed tannins. Yesterday I slightly mentioned the condensed tannins, that is polyflavonoid tannins, polymer of the flavonoids, and chemistry, biological significance, and commercial uses of condensed tannins. Yes, stilbenes, you can see they are, uh, these are the structures of different stilbenes. Here you can see, like flavonoids, they have two aromatic rings, but there is a difference. Difference is here. Here you see, in case of flavonoids, a ring and a B ring and a C ring, there was C3, but here you can see C2. So stilbene and flavonoids structurally, here is the main difference. So they are obviously phenolic extractive, can be extracted from the plants, found in the hardwood of hardwood and softwoods of the plants, mostly hardwood of the plants. They are colorless or yellow unsaturated crystalline hydrocarbon compounds containing C6, C2, C6 backbone. So if you are asked what is the backbone of stilbenes, their backbone is C6, C2, C6. Here you see C6, that means one benzene ring connected to another benzene ring with two carbon chain. So stilbenes are basically hydrocarbons, obviously phenolic compounds, and they are prevalent in many plants, including the pinus. Uh, they cause light-induced darkening in wood. You have seen in case of wood or timber, which we use for furniture, sometimes darkened color in the wood is indicating the presence of stilbenes and obviously the quality of the wood. The darkened color hardwood of the plant is good for furniture because they are powerful in protecting the insect and other microorganisms. As a result, the wood lasts for longer time in the furniture. So cause light induced darkening in wood. That means darkened color in the wood, hardwood of the plant is basically the principle is the light induced conversion of the stilbenes. Often two aromatic rings. I mentioned two aromatic rings. Here you can see one aromatic ring, another aromatic ring, and occasionally more, sometimes more aromatic ring can be present, but two aromatic rings minimum. Presence of double bond linkages. Here you can see the presence of double bond linkages unsaturation, and naturally present most often in transform. Cis and transform in the chemical structure is very important. Cis and transform chemical structure, their properties are also different, although molecular weight are same. So they remain as transform most in stilbenes. So this is 4-hydroxystilbene. This is pinosilbene, and this is pisade. So these three are the examples of stilbenes. Stilbenes biologically very important, and they have significant biological activities. They improves decay resistance, reduced fungal activity in the wood, improves insect resistance. Stilbenes in leaves reduce insect feeding, and also reduces animal damage, because secondary metabolites are produced by the plants for their own protection. So stilbenes can protect plants from the microorganisms, for example fungi, insect, as well as from the animals. Pinosilbene containing leaves have been shown to suffer less damage from the snowshoe hair consumption. Snowshoe hair is an animal, and less it eats the pinosilbene containing leaves. So that means animal eating also resisted by the stilbene. So stilbenes and pulp and pepper problems, this is very important scientific discovery, because stilbenes poses some problems in making the pulp and pepper. Another problem is effluent issues. Chlorinated stilbene, during pepper making, chlorination is a process. So chlorinated stilbene derivatives cause problems in effluents, especially fish are affected at low levels. Very low level concentration in the effluent, if disposed to the water bodies, can affect the aquaculture, fish killing. So stilbenes environmentally in pepper industry, they are problematic, not only inhibiting the pulp and pepper making process, but also environmentally chlorinated stilbenes are problematic. Pulp problems, let me discuss a little bit. Discovered in 18th century that certain trees, pine and others, cannot be pulped by the acid sulfate process. Pulp making process, one important step is making the acid sulfide, and in that process, presence of stilbenes in the plant materials causes the serious problem. So scientists discovered that that problematic culprit is stilbenes, which cause lignin cross-linking reaction. So what stilbenes do? Stilbenes, cause lignin cross-linking reactions, and condensation taken place. As a result, pulp, a good quality pulp cannot be produced, and from that pulp, pepper also cannot be produced. So stilbenes are chemically disturbing for making the for the pulp industry. If a plant contains higher amount of stilbenes like pine, they are not useful for pulp making and pepper industry. Flavonoids already I discussed in yesterday as lecture. They are ubiquitous monomeric natural product present in various kinds of plants and serve many roles in plants: protection, coloration, other unique roles yesterday I discussed, and this is a structure of a catechin. Yesterday I showed you. So flavonoids general structural formula I repeated several times. They are C6, C3, C2, C6. So quite different from the Stilbenes. Stilbenes has C2 here. So C6, C2, C6 is are Stilbenes, uh, skeleton, and in case of flavonoids C6, C3, and C6. So polyphenolic compounds found in softwoods and hardwoods of plant are called flavonoids. So they are polyflavonoid, contain lots, many, hydroxyl group. Polyphenolic compounds composed of diphenyl propane units. This is called diphenyl propane units. This unit is unique in the flavonoids. 8 structural classifications. They have 8 groups of classes of structures. We shall discuss and show you the structures, which is very important. Flavonoids differ through substitution on the rings, typically hydroxyl or methoxy groups, and typical to have hydroxyl on position. Yesterday I mentioned 5 and 7, 5 and 7 carbon easily hydroxylated, as well as 3 prime, 4 prime, and 5 prime are also hydroxylated and are also present as glycosides. Flavonoids can be present as glycosides. So what are glycosides? Glycosides means if a flavonoid is connected, contains glucose or any other sugar, they are called glycosides. So hydroxyl group can be substituted by a glucose molecule, and they can be glycosidic flavonoids. So these are the 8 groups of flavonoid. Classification of flavonoids includes flavones, flavonols, flavonols, flavonones. Here there should be one N. There is a spelling mistake. So flavonones, N-O-N-E-S. Flavonols, catechin, leucoanthocyanidins, chalcones, and orones. So these are 8 groups of flavonoids, and there are huge structural differences in each groups, and lots of compounds have already been discovered. So flavonoids, representative structures of flavonoids. Yesterday I showed you several. So chrysin, texifolin, pinosembrin, pinobunskin, catechin, quercetin. These are the important classes of groups or structures of flavonoids, examples of representative chemical structures of flavonoids. So flavonoids biologically very significant. Not all flavonoids have same functions in plants. They have various functions. For example, antifungal. Yesterday someone asked me whether flavonoids have any antimicrobial activities. Yes, flavonoids have antifungal activity mostly, and obviously some inhibitory activities against other microorganisms, and they are uh, inhibitory against the insect feeding. Some feeding deterrents and some stimulants are flavonoids, insect feeding deterrents, and some insect stimulants of feeding. Antioxidant, yesterday I discussed in detail the activity of antioxidant activity in flavonoids. This is why dietary flavonoids are very good for the health. And color, bright colors in the petals of the flowers, fruits, and other plant organs, they are basically flavonoidal compounds, and they are useful in the dyeing industry. Flavonoids location in plants. Where in the plant flavonoids are found in the heartwood of most trees, referred to as most ubiquitous monomeric natural product. often responsible for coloration and found in lesser amounts in the sapwood also. So mostly found in the hardwood, but sapwood also contains, but they are also present in large amount in the bark, the skin of the plant bark, and they are also found in the roots. That means sapwood, hardwood, bark, and roots. That means every plant organ contains flavonoids. Even plant leaves also contains the flavonoid. Certain flavonoids play role in trees' interaction with the beneficial organisms. Some flavonoids, they are very important in plants' communication with beneficial organisms. For example, you know, rhizobium legume symbiosis. Rhizobium plants attract the leguminous plants attract the rhizobium bacteria to their roots for making the nodule. So some flavonoids like luteolins and other luteins and other flavonoids attract specific rhizobium to their roots. So flavonoids play a role in trees' interaction with the beneficial organisms like nitrogen fixing bacteria. Leaves green tea can contain up to 30% of flavonoids. So if you are asked the example of a plant leaves which contain high amount of flavonoids, the most important example should be the leaves of tea. And green tea contains 30% up to 30% flavonoids in their dry weight. This is why very good for our health. They offer us, good, to boost our immunity because flavonoids have antioxidant principles. Flowers' color is often due to the flavonoids, color of plants and other organisms. So flavonoids have medicinal uses. Higher flavonoid content in diet reduces chances of heart disease, strengthens capillaries, and dilates blood vessels. So keep our heart functional. And some of the flavonoids also help us to keep free from the stroke. And some flavonoids have anti-cancer principle. All types reduced. Some flavonoids reduce the cell division of the cancer cells, and lots of study have been done. In case of soybean, flavonoid genistein and diargine, they are powerful anti-cancer as compound, as well as they have the powerful estrogenic activities. So keep the human, especially the female, after the age of 50 or after the menopause, if they take green or soybean seeds, they can get genistein and diargine and keep them younger, and very good for the health, for the skin, as well as for making the hormonal balance in the body. So flavonoids also important in pulp and paper problems. Flavonoids, especially taxifolin flavonols, also cause problems in sulfite pulping. Sulfite process is impaired or inhibited or disrupted by the flavonoids. Flavonols are oxidized by bisulfite to flavonols, producing thiosulfite. This is the real problem. Thiosulfite causes lignin condensation reaction. So flavonoids also associated with lignin condensation during the pulping, and is a real problem. So taxifolin flavonols react with the sulfuric acid and produce the quercetin and the sulfide, bisulfide, and really causing the problem of lignin condensation reaction. So condensed tannin, another very important group of flavonoids. Condensed tannin. Condensed means several monomeric flavonoids are polymerized So condensed tannin we can name in other ways the polyflavonoid tannins. Uh, Polyflavonoid tannins, condensed tannins are formed through the polymerization of flavan-3-ol, catechin, and flavan-3,4-diols leucoanthocyanidins. So catechin and leucoanthocyanidin, they specially polymerize these two structure and join together and make a polymer, and they are called condensed tannin. In tree, polymerization through acidic enzymatic non-oxidative coupling taken place, and polyflavonoid tannins can be 2 to 50 units. So 2 units of flavonoid can join together, make the smallest condensed tannin, and the unit can be unit of flavonoids in a polyflavonoid can be up to 50, typically 2 to 8. So linkage as can be through a variety of sites. So they can be very complex structure, and polyflavonoid tannins are very important in plants, especially condensed tannins. They impair, uh, iron absorption in the body. So tannins are problematic. Dietary tannins in the animal fish feed or human feed is problematic for iron absorption. So we have to be careful whether the presence of condensed tannin, the level of condensed tannin presence in the dietary food of animals, as they impair the iron absorption and other creates other problem in nutrition. So condensed tannin, I mentioned the structure. You see how they are connected to each other, and here 3 units, typical condensed tannin structure, catechin and leucoanthocyanidin. They bind to each other and form this condensed tannin. This is the how to say typical structure. But here you see it can another unit can be connected here, another unit can be connected here and make a big polymer. Condensed tannins location in the plant, western hemlock bark. Bark is a very thick bark of plant, contains condensed tannins. For example, western hemlock 18% tannins of the bark. In our country, Lania coromandelica, jiga or kafila tree, which is a wild tree used for fencing the farm and houses. Their bark also contain high amount of condensed tannin. Wood contains very low amount of condensed tannin. So bark is important organ of the location of the plant's condensed tannin. So western hemlock and quebracho, another in wood, tannins found in the heartwood. But in case of quebracho, heartwood is almost condensed tannin, majority 80% located in basal lumina of the plant tissue. Tannins deposit in dead cells starting at CML and ending at the secondary wall. And in case of Lania coromandelica, I mentioned in Bangladesh it is abundant. A fencing plant, kafila or jiga tree, their bark contain 13% of tannin. This Lania I studied a lot, and in some lecture later on in on different bioactive compounds, I shall discuss the biological activity and chemical properties of Lania bark condensed tannin. Mimosa, another plant, also produces mimosa tannin. So these are, if you are asked to name some important plants that produces condensed tannin, quebracho, lania, mimosa, western hemlock, you can name them. So condensed tannins, they have biological significance, as I mentioned before: protection of plants against insects and animals, bad test, astringency, beta test, astringent test, beta or you can say beta test inhibit the insect and animals to eat the plant. Animals tannins reduce digestion of the food. So presence of condensed tannin at higher concentration reduce the digestion of the food by the animals, including the fish. Toxic to bacteria, so it is good quality. Pine, calasses created by fungal invasion. Tree forms calasses as protective tissues. So some calasses are produced in the due to the condensed tannin, and calasses are also protective tissue. Calasses contain high levels of tannins, for example, 50 to 80%. So condensed tannins have biological activity. In case of plants, they are protective agents and inhibit the bacteria, fungi, fungal invasion, as well as inhibit the animal feeding. So uses of condensed tannin, Watol and Quebrechu used in the tanning of the animal hide and skin. So tanning industry, they are very useful. Leather tanning, 10,000 plus year old industry, very old. So people have been started to use Quebrechu and Watol and other mimosa tannin in leather tanning even 10,000 years before. Vegetables tannin and chrome tannins interact with potential in hides and adhesives. They are also can be used as adhesives, biocides to kill different microorganisms and other organisms, and chelation, micronutrient fertilizer. In some micronutrient fertilizer, as tannin can chelate, so micronutrient fertilizer can tannins are used to for the retention of the fertilizer in the soil and rhizosphere for longer time. So it increase the efficacy of the fertilizer for longer time to stay in the rhizosphere. Mineral flotation, this is another principle of the use of the condensed tannin. So thank you very much. This is all about today's lecture, and now we shall discuss a little bit. So you are welcome to ask the question. So unmute your microphone and then ask the question.
Sir, Assalamu alaikum, Jinan.
Yes, Jina.
Sir, is Stilbenes is less important for plants or other uses than flavonoids, as it is only found in the hardwood of the softwoods, and uses is less.
So your question is whether the presence of stilbenes are limited in plants as well as its use is also limited compared to flavonoids. Yes, flavonoids,
Yes.
their uses are robust. You see the pollination. Pollination is triggered by the flower colors as well as flavonoids. They are the major compounds involved in antioxidant activity in the plants to detoxify superoxide produced in the plant cells. But stilbenes, they are also phenolic compounds. In some plants, they are very important. For example, in case of tea plant, tea plants, flavonoids are present high quantity, but in pine plant, in the hardwood, stilbenes are present. Pine and some other plants, especially the timber yielding plant, the hardwood means the internal portion of the wood, center of the trunk. They are rich in Stilbenes and helping us to make different furniture because the wood quality dramatically increase if they contain higher amount of steel beans. So steel beans industrially important because uh, in wood and furniture, especially the quality of timber is highly improved by the presence of steel bean. But obviously compared to flavonoids, their functional rules as well as economic importance, industrial importance, steel beans are less than the flavonoids. Flavonoids are very robust group of phenolic compounds. Thank you.
Thank you, sir.
Welcome. Next question. So 25 students are now attending the class. So next question. We discussed on alkaloids, terpenoids, and phenolics compounds, especially the secondary metabolites, their structures, functions, biosynthetic pathway. Later, we shall discuss how can we discover these compounds from the natural sources. That means the analytical process, industrially as well as in the laboratory. How can we, study those natural products, how can we discover new natural products in the laboratory, as well as how can those compounds be used in the industry, including the pharmaceutical industry in the later class. Any more question? No? Have you understood thoroughly the lecture? Do you have any problem in participating the online lecture or getting the information correctly? Do you have any problem?
No, sir.
We shall discuss a little bit if you have serious problem. No?
No, sir.
Any question? Last chance. So how can we take the midterm exam? Can we set the date for midterm examination? Is it possible for you to do the midterm examination in this weekend? First midterm. So that would be open book, question. But what you need to do, you will get maximum, 12 hours time to answer the questions that I will, share with you through the Facebook for the midterm one. And then you can prepare the answers, obviously not handwritten. You have to type the answer in Microsoft Word format and then send me through email. So I shall set the time, and you must do before the deadline. If I offer you 12 hours time or 6 hours time, so within that time you have to answer all the questions and then send me through email attachment. So is it okay if we take the midterm one exam in the weekend, for example, coming Saturday?
Assalamu alaikum, sir.
Walaikum salam. Is it okay coming Saturday for midterm one?
Yes, sir. I think it's okay, sir. And everybody.
Do you have anyone have any problem? So it would be open book exam. So you can check the internet as well as your notebook, my lecture, anything else, and you make the answer and prepare the answer, compose in Microsoft Word file, and then send me with your name and registration number, student ID. The syllabus should be, condensed tannin, and the first primary lecture, the introductory lecture, alkaloids and terpenoids. Is it okay? Introductory lectures and terpenoids and alkaloids. And next second midterm we shall include stilbenes and flavonoids. Okay.
Yes, sir.
Yes, sir.
Yes, if it is okay, then I shall, prepare the questions for you. So be prepared for the Saturday exam. So you shall get, several hours of time, maybe 6 hours or 12 hours, I'm not sure, depending on the question type. And you can answer the questions and then send me through email attachment.
Okay, sir.
Okay, thank you then, and have a good day. And please take care from the pandemic COVID, and be safe and be healthy.
Yes, sir. Assalamu alaikum.